Isolation of peripheral blood lymphocytes by fecal hyphae gradient. The first step in studying lymphocytes is to isolate them so that their behavior can be analyzed in vitro. Human lymphocytes can be isolated most readily from peripheral blood by density centrifugation over a step gradient consisting of a mixture of the carbohydrate polymer fecal and the dense iodine-containing compound matrizamide. This yields a population of mononuclear cells at the interface that has been depleted of red blood cells and most polymorphonuclear leukocytes or granulocytes. The resulting population, called peripheral blood mononuclear cells, consists mainly of lymphocytes and monocytes. Although this population is readily accessible, it is not necessarily representative of the lymphoid system, as only recirculating lymphocytes can be isolated from blood. A particular cell population can be isolated from a sample or a culture by binding to antibody-coated plastic surfaces, a technique known as panning, or by removing unwanted cells by treatment with specific antibody and complement to kill them. Cells can also be passed over columns of antibody-coated nylon-coated steel wool, and different populations differentially eluted. This technique extends affinity chromatography to cells and is now a very popular way to separate cells. All these techniques can also be used as a pre-purification step prior to sorting out highly purified populations by FACS. And that's isolation of peripheral blood lymphocytes by fecal hyphae gradient.